Welcome to everyone to the Mountain States Regional Genetics Network Genetics Summit 2023 with our theme this year of peak performance. We're on track number 4, the K, which stands for karyotypes to knockouts. It's my pleasure to introduce the first session of this track entitled At the Forefront of Genomics, Making Genomic Medicine a Reality. And we're so excited to have Dr. Eric Green with us, who's the director of the National Human Genome Research Institute. And his full bio is available at the attendee website under the bios tab. Please reference this continuing medical education slide and disclosure statements, as well as the learning objectives for this presentation today. And Dr. Green has no disclosures. With that, Dr. Green, the floor is yours.
Okay, well, thank you very much for that wonderful introduction. What I wanted to really tell you about today is what I think is the cutting edge of certainly human genomics, which is the reality that genomics is now becoming relevant in mainstream medicine. Before I take you there, just to reiterate, and you just saw this on previous slides, I have a couple learning objectives, but it's all about the storyline of genomics over the last 30 plus years, leading to the current situation where we are really implementing genomic medicine. I don't have any disclosures, and the major reference that I'll get to in my talk near the end is this latest strategic vision that the Institute has put out. So let me remind you how young this field of genomics really is. Many of you think a lot about genetics, but genetics is a much older discipline. Genetics, the word and the field, was coined and created just slightly after the turn of the century, around 1903, 1904. But it wasn't until the latter part of the 1900s, in 1987, that the word genomics was actually coined. The word was coined and first put into the medical and scientific literature in this lead editorial of a brand new journal that was created called Genomics, where the editors described this newly developing discipline of genome mapping and sequencing. And the reason why a new field was created and a new name was coined for the field of genomics was because by the late 1980s, it became very clear that the community was getting ready to move forward on a very big, audacious project that was going to really launch the field in a very significant way. And that project, many of you are familiar with, was, of course, the Human Genome Project. The Human Genome Project began in October 1 of 1990. I got involved in it from the very beginning, then as a recently graduated MD PhD student, and got to participate on the front line of the project from the very beginning to the very end. And of course, there were many smaller goals in the Genome Project, but the signature goal, which marked, once it was completed, the end of the Human Genome Project, was generating that first sequence of the human genome, laying out the roughly 3 billion letters of G's, A's, T's, and C's that constitute a representation of a human genome. And of course, those who are familiar with the history of genomics, and in particular who followed along during the Human Genome Project, might remember, for example, seeing the reporting of the draft sequence, for example, of the human genome, featured both in Nature and in Science. The Nature article in particular was featuring the work of the Human Genome Project. The Science article was featuring the work of a competitive private sector project that had also generated a draft sequence of the human genome. And in fact, if you were paying attention back in 2021, you might have remembered that those same two journals featured once again on their cover to commemorate the 20th anniversary of having a draft version of the human genome sequence in hand, commemorating those 20 years that had passed since that draft sequence had been generated. But much more poignant was the fact that this year, we were able to celebrate not the 20th anniversary of the draft sequence of the human genome. We celebrated the 20th anniversary of actually completing the entire Human Genome Project, including generating that first nearly complete sequence of the human genome. And so looking back 20 years ago, it's an interesting thing to reminisce about, is that we've now had 20 years with the sequence of the human genome laid out before us, and with that has provided incredible opportunities to capitalize on some of the reasons why we went and did the Human Genome Project and why we thought it was so important to generate a human genome sequence and to complete the goals of the Human Genome Project. Now, let me remind you, if you don't remember quite then or didn't quite appreciate it, the real rationale that convinced the US Congress and several other major funders around the world to support the International Human Genome Project was the idea that we appreciated the fact that the way we were practicing medicine and the way we were understanding human biology was based on average people. That all medical decisions were based on what worked for the average individual, and all biological insights were looked at sort of on average for human physiology. But none of us is average. Every one of us is unique. And we knew that some of our uniqueness, not all of it, but some of our uniqueness was scripted in differences in our DNA and our genomes that made us either more or less susceptible to a certain disease, or maybe protected us from certain diseases, or gave us other traits that were medically relevant. But we had no insights what those were because we had no way to peer into an individual patient's genome and actually use any of that information as part of their clinical care. If, however, we could actually have the human genome sequence in front of us and the tools and technologies to be able to understand the genome and to be able to go in and interrogate a patient's genome to look for differences in their genome compared to other people, maybe someday genomics could become a relevant part of medicine. So when the Genome Project ended 20 years ago, it wasn't terribly surprising that the idea of putting together the concepts of genomics and medicine made a lot of sense. That is why the popular press and the scientific press both started to feature the phraseology of genomic medicine on the covers of these prominent publications. And we got very excited about this, the idea that one day maybe we will see genomic medicine become a reality. There was a problem with that 20 years ago in that we really didn't know what that meant. It really was a rather blurry concept. It was something that we could type out, but we really couldn't bring it into focus. We could define it, and we did define it as an emerging medical discipline that needed to be developed that involved using genomic information about an individual as part of their clinical care. In other words, don't treat a patient as if they're average, tailor their medical care based on their unique genomic information. But of course, to actually realize genomic medicine, it was very clear that we were going to have to think about ways that we were going to take a more personalized, a more individualized, and a more precise approach to actually practicing medicine. And these are these related terms I show at the bottom here that, are roughly synonymous with genomic medicine. They tend to be words that include other aspects of our personal life, our social and physical environment. I tend to use the phraseology genomic medicine, but it's roughly synonymous with these other phrases you see on the bottom of the slide. How are we going to get to genomic medicine someday? Well, like the Genome Project, it was going to be a journey. It was going to be a journey of undefined steps, but it was going to be a journey that was going to be long and challenging and have as its starting line the Human Genome Project being completed. And as its finish line, something that was akin to realizing genomic medicine, recognizing that there were going to be many steps along this journey, only a subset of which did we even know what those steps were going to be. We also recognize that, like the Human Genome Project, if we just did one step at a time, we could just cross it off our list, and then we could inch our way closer and closer, eventually knocking down all the steps and realizing genomic medicine. But this was not going to be a sprint. It was going to be much more analogous to a marathon. And a marathon's a good analogy because it also was going to involve a lot of people, a lot of disciplines, a lot of countries, a lot of institutions working together, running together, probably what's going to take many decades to fully realize. But if we could have a strategic approach for knocking down each of these steps, making this progression a reality, we should be able to see genomic medicine realized at some point in time. So that was our approach for the last 20 years. You may say, well, what are the steps that were critical for this? Now, here in this talk, I don't have time to march through all the progress that's been made in 20 years. So if I had to condense 20 years of progress into about 6 or so featured highlights, I would say it was as follows. And here again, I'm condensing 20 years of progress down into about 6 PowerPoint animations. The first one was what I already told you. The first major step towards realizing genomic medicine was actually sequencing the human genome for the very first time. That was accomplished by the Human Genome Project. We could scratch it off our list. The problem with human genomics 20 years ago was that we got the first sequence of a human genome, but it cost us over $1 billion to get that sequence. And as all of you appreciate, that is a very expensive clinical test, not one realistic for most patients. But what we were able to do in the last 20 years is to develop a series of better and better and better technological innovations that allow us to reduce the cost of sequencing a human genome by more than 1 million-fold. We've gone from $1 billion to sequence a genome to under $1,000 to sequence a genome. And now genome sequencing is cheap and the amount of data you can generate is amazing. And that is no longer rate limiting. With cheap DNA sequencing methods, we didn't have to just have one human genome sequence. We could use those methodologies that have now sequenced millions of human genome sequences. With that, we have learned about the 3 to 5 million variants that each of us carries in our genome compared to your friend or your neighbor or some reference sequence. And so through the sequencing of millions of people's genomes, we are getting more and more information about how our genomes vary across different human populations. And so human genomic variation is much clearer to us now, but it was an important thing to help us define. But of course, you don't want to just know about variation. You need to know about how does variation influence genome function? How do misspellings in DNA in some way influence biology, in some ways might be medically relevant? And here you must understand how the human genome works. And while we are a far way from knowing all the intricacies of the human genome, we have made profound advances in understanding how the human genome functions. We now have a pretty good inventory of our 20,000 genes or so. We're increasingly understanding the regulatory sequences involved in dictating when, where, and how much those genes get turned on, and so on and so forth. And while, again, it'll be decades until we've totally finished understanding how the human genome works, we've made a lot of progress in 20 years. With the ability to sequence cheaply, have knowledge about genomic variation, and how that variation is increasingly becoming clear how it influences genome function, we're able to now begin to look at different genetic diseases, rare diseases and common diseases, and be able to see which of the variants that are conferring risk or are overtly causing a particular genetic disease. And while, again, we've seen the greatest advances with rare diseases, but we are learning a lot about common diseases, and we are beginning to understand more and more about the genomic architecture that underlies the genomic risk that influences common diseases. So this progression of 20 years, and again, I'm being very superficial here in this slide, but it gives you an appreciation of a logical progression of steps that are first very technological, then very biological, then a little more translational as we're dealing with human disease. But these first set of steps really didn't deal with the practice of medicine. It was still up to the translational part, but not the clinical part. But what I really want to emphasize in this talk is that we have started to see a final step in that progression, in that journey towards genomic medicine, because vivid examples of genomic medicine are now emerging for the very first time. And this has really happened, I think, faster than some of us anticipated. The reason these things have come on the scene is because generating a sequence of a human genome is actually quite routine. You could do it for less than $1,000, and you could at least interpret some of the differences that you find in every genome compared to any other genome. And oftentimes, you can figure out which of those might be medically relevant for the condition that the individual patient is presenting with. And so what we have is genome sequencing becoming mainstream in medicine, whereby we are figuring out which variants individual patients have by sequencing their genome, such as that on the right, comparing it to a reference genome, a known high-quality reference genome, and simply developing lists of genomic variants that exist in the patient that don't seem to exist in a reference. And so starting about 5 years ago, it seemed that everything was coming together in this progression from the base pairs of the Genome Project towards the bedside of patients. We had cheaper sequence, knowledge of genomic variants, understanding how the human genome works, and more knowledge about the genomic basis of human disease. And what that meant were these vivid examples started to come to the fore where genomics was actually being used in medicine. And while they're the earliest examples, they were wonderfully helpful because they finally brought clarity to what genomic medicine was going to be all about. And I think that clarity has helped us see through these earliest exemplars what is going to be like to actually have genomics be part of medical care. Now, the examples I'm going to give you were not coming about through some grand organized plan of which things we were going to do first. They really were all opportunistic in how they bubbled up. They represented unique diagnostic needs or unique technological applications where there was a clinical need. And in each case now, you are seeing real advances medically because of bringing genomics to bear with individual patients. So I'm going to give you 4 examples. This is going to be the main part of my talk. And then I'm going to take you a little bit more to the future when I'm done. So what are the areas where genomic medicine is being implemented? Well, first and foremost is in cancer. And here I would just say, I think the greatest impact of genomics, especially in the near term, is going to be in the cancer field, in the diagnosis and management of cancer patients. I think many of you are fully appreciative of the fact that cancer is a disease of the genome. You have normal cells that pick up mutations and aberrations in their DNA that eventually become tumor cells. But we can read the DNA of tumor cells just as easily as we can read the DNA of the normal cells from that patient. We can compare them and we can know exactly what has gone wrong in the DNA of those tumor cells. And what that is doing now is augmenting what has traditionally been very important in cancer, which is cancer histopathology, pathologists looking at it under a microscope at a piece of the tumor. But now we augment it by sequencing the genome of that tumor from that patient and making a map, if you will, a signature of the DNA changes that have taken place in that tumor, not in the average patient, but in that patient. And so morphology is being augmented with genomic signatures, and with that is more powerful than either one alone. And there's been very interesting changes to our paradigm when it comes to cancer care. One of them being is that a genomic signature of a tumor is often, perhaps even always, provides more valuable clinical information than the tissue of origin. In other words, if you have a breast tumor and you have a liver tumor and they both look the same genomically, that is more important to know than what tissue it was cut out of. And in the past, we focused much more heavily on what tissue the tumor came from. Nowadays, we realize a lot of the important prognostic and management issues and information coming out of these analyses comes from the genomic signatures. So it is fully expected that cancer genomics is going to be mainstream. A variant of this, besides sequencing the DNA of a tumor, is something known as a liquid biopsy, because tumors, when they exist in the body, do have their cells breaking down and shedding out cell-free DNA. And cell-free DNA can be analyzed if you draw a little bit of blood from that patient. You could use these super sensitive methods of DNA sequencing to analyze that cell-free DNA. And basically, instead of having to do a standard biopsy for human tissues of a tumor or to search around the body if there exists a tumor, you can just use the fact that the DNA is being released in the bloodstream and use these highly sensitive DNA methods to detect and analyze that cell-free tumor DNA that you've accessed through a simple blood draw. That could be done in surveillance in healthy patients looking, and you're hearing about this more and more, of surveillance to look for the first detection of a tumor, or it can be used post-treatment to see whether a cancer comes back. So a lot of people get cancer, get treatment, they think they're cancer-free. One way to detect early cancer is by monitoring for the presence of cell-free DNA. And you even know what you're looking for because you know something about the DNA changes that took place in that tumor, in that patient. Notice how individualized all of this is becoming increasingly because of genomics. So I think cancer, it's going to be a big deal. We could fully anticipate that. A second area is going to be rare genetic disease diagnostics. Here, this has just been game-changing. You know, the fact of the matter is more and more we're seeing patients present, such as children often, like with a cardiac defect or with a developmental delay. And in the old days, you would work them up and you do all sorts of diagnostic things. But nowadays, it actually is cheaper. Just sequence their DNA, get their history, but then also collect their DNA, sequence the DNA, compare it to a normal sequence, a reference sequence. And with that, you get a lot of information. This is being done probably 20 to 30,000 times a year in the United States, probably double that worldwide. It is just a standard diagnostic tool early on in the workup of someone who is suspected to have a genetic disease. There are also a lot of people, both children and adults, that have gone around for years with undiagnosed conditions. And the idea of having genomics be brought to bear in these situations have led to the creation of undiagnosed diseases programs, or nationally, we have an undiagnosed diseases network, where sometimes you have patients that have failed to be able to find a diagnosis for through standard diagnostic workups. You work them up one last time. That includes a genomic analysis. And these programs have been extremely effective at identifying the genomic basis for not all the patients, but some of the patients that had failed medical surveillance and diagnosis in many sites, in some cases for decades. Another setting where genome sequencing is proving to be incredibly valuable is in the setting of acutely ill newborns in neonatal intensive care units, where sometimes you're dealing with days before that child might perish. And through a series of innovations that have taken place, we can now sequence a newborn's genome in a matter of a day or two. And in many cases, figuring out what is wrong with that child, even when that child is completely a mystery to the neonatologist. And in some cases, there are interventions that can take place that could actually save that patient's life. And you don't need very many of those circumstances to convince you that rapid genome sequencing of acutely ill newborns is a worthwhile thing. We're seeing it more and more in neonatal intensive care units around the United States, and even now increasingly around the world. So rare genetic disease diagnostics by genome sequencing is becoming mainstream. The number one genomic test in the world now is actually from the prenatal side of things, prenatal genomic testing. Here, of course, is the use of non-invasive prenatal testing. Traditionally, we would do prenatal testing before genomics by methods that access the fetal DNA that were quite invasive, like amniocentesis, and we'd be able to look at the unborn child's chromosomes and detect things like aneuploidy. But, you know, those are costly, they're dangerous, they're uncomfortable, they do risk the pregnancy, and we don't have to do that anymore. Because it turns out that with genomics now, we can access the small amount of fetal DNA that naturally gets shed from the placenta, and the placenta represents fetal DNA. And as an alternative to invasive procedures, we just sequence the cell-free DNA, and we can get that same screening information to detect aneuploidy, just like we could do if we use more invasive methods. The reason I'm particularly excited about this is because there's 5 to 7 million pregnant individuals will get a non-invasive prenatal test done this year. Imagine 5 to 7 million individuals getting this test done because it's turned out to be so inexpensive, so incredibly effective, and so non-invasive. And the fourth thing that is here and now, although we expect it to grow substantially in the future, is in the arena of pharmacogenomics. Big word, pharmacology and genomics. It just simply reflects the fact that people respond differently to actually everything, but just like roller coasters, I'm the guy in red. A lot of people are like the kid in white. But just like in roller coasters, we respond differently to medications. Every one of the medications shown here, they all work. Otherwise, the pharmacy wouldn't prescribe them. They just don't work for everyone. And we have increasingly realized that actually, in most cases, they don't work for most people. It's incredibly imprecise. Shown here are 10 of the most commonly prescribed medications. And for every individual shown in blue where the medication works, or all the individuals in red where the medication doesn't work. This is not particularly precise, but we can do better. We can get the right drug for each individual, not by inventing the drug for each individual, but by selecting the best drug among a list of drugs. There are, 10, 20 medications for hypertension. There are, 5 to 10 medications for depression. There's 6 to 8 medications for asthma. And doctors tend to be very imprecise on which drug they pick for which person. But we are figuring out we can do better because we are learning that if you take groups of patients who have the same diagnosis through genomic screening, you could figure out which ones happen to have DNA changes in key genes that influence the drug metabolism of that particular medication. And by doing the pre-screening, you could stratify these patients into the green group, which the drug, the drug, might be very appropriate for them, or not be appropriate for them, whether it's green or red or yellow or so forth. You want the drug not to be toxic, but you also want it to be beneficial. So you actually want to be in the group on the bottom right. And so you can see that you have different categories, and what you're trying to figure out is low toxicity, highly beneficial. And we can do that. And that's becoming more and more mainstream, where one size does not fit all. We simply stratify based on your genomic signature, and we get the right drug to the right patient at the right dose. This is happening now for a handful of medications, and it's fully expected that that list will grow in the coming decade. And so when, is that really true for pharmacogenomics? Well, there's been studies that have looked at it and said that out of all FDA-approved medications, 7 of them are known to be influenced by a gene that when variants are in it, influence that metabolism of that. And that those set of prescriptions account for about 1/5 of all prescriptions in the United States. And that was 8 years ago. It's probably even higher than that. And shown here is just a company. It's not a company I'm endorsing, but just one of many diagnostic companies that lists many, many different genomic tests that they will do in genes that are relevant for pharmacogenomics. And to give you a flavor, these are very commonly used drugs. So look at these drugs here. All of these drugs have known pharmacogenes associated with them, where if you know what the variants are, you might be able to figure out whether it's a good drug or a bad drug for an individual patient. Again, very common drugs where this is highly relevant. So the bottom line is pharmacogenomic testing is now appropriate for a small subset of prescription medications, and we expect that subset will grow in the future. And there's one last area I just want to point out because you hear about it, and certainly there are companies that are offering this direct to consumer, not involving a patient, and that has to do with risk predictions. Can you predict which people early in life might be more susceptible to sudden cardiac death, or to seizures, or to arrhythmias, or to diabetic states, and so forth? And so you might hear the phrase polygenic risk score. And this is not by understanding the underlying biology, it's just by developing correlations between sets of genomic variants and whether or not that puts you in a high risk, a medium risk, or a low risk category for getting a particular disease. I don't think this is ready for implementation in mainstream medicine yet, so I'm going to put some question marks here. It is, however, an active area of research that I think we should watch over the next decade. And at the National Human Genome Research Institute, where I work, we are investing a lot of money into studies that are going to look at whether or not we can develop genomic risk tests that might predict future onset of especially more common genomically complex diseases. So stay tuned about polygenic risk scores. So that's what I wanted to tell you about sort of very quickly where we've been in genomics, the Human Genome Project, the last 20 years, and in particularly with an emphasis on how we are now making genomic medicine real. But there's also the future that think about, because that's what we think a lot at my institute at NIH, is how can we enable a future? How can we make things go faster? How can we make genomics even more mainstream in medicine? And I will also tell you that while I have quickly told you this incredibly impactful story of what's happened in human genomics over the last, say, 30 or 35 years, it hasn't been a straight line. It's been a complicated journey with many twists and turns, and everything is changing about it all the time. Where we were 20 years ago is so different than where we are now. I was really moved by this piece that came out in The Economist earlier this year in an article that was celebrating the 20th anniversary of the completion of the Human Genome Project, where they talked about how no part of biology has been untouched by the consequences of the Genome Project. And genomics has really become a framework for biology, just like the periodic table forms one for chemistry. I mean, genomics touches everything. And I think that's been a very transformative aspect of our field, that everywhere you look, genomics is touching in the life sciences, and increasingly that is the case in medicine. It actually led us to change our self-identity. We have a new organizational mantra we started about just before the change of the decade, where we're no longer about all of genomics, we're just about the forefront of genomics. That's what our institute does in leading genomics research in the United States. And this was sort of the battle cry and the organizing mantra for us developing our latest strategic vision, which we do about every 8 to 10 years. And in October of 2020, we published this new strategic vision. This is the paper I put as the reference. There's one paper to please read relevant to my talk. It would be these 10 pages in Nature, where we talked about our vision for improving human health at the forefront of genomics. And we talked about that what was going to be needed to accomplish it was not just doing a lot of big research projects or designing a lot of cool technologies, but rather the forefront of genomics meant that we needed to take responsible stewardship of the field. And that responsible stewardship needed to be expressed in a number of domains, 4 in particular. One domain was taking responsible stewardship for the guiding principles and values that undergird the whole field of genomics, thinking about things like diversity and equity and social justice, thinking about things like of importance of open science and the importance of data sharing and so forth. And these are things that need to be articulated and built upon because it influences everything we do in genomics. Being a responsible steward of genomics at the forefront of the field meant developing and maintaining a robust foundation for genomics. Think about all the data resources and the software. Think about all the technologies that need to be nurtured and data standards that need to be associated with it. Thinking about tools for improving literacy in genomics across many different sectors. That's the foundation for genomics that everyone relies on. And we are going to be responsible stewards of maintaining and enhancing that foundation. Part of the stewardship responsibility at the forefront of any field is figuring out what is impeding progress in that field and breaking down those barriers. 20 years ago, it was the cost of DNA sequencing, and we broke down that barrier by developing new methods for DNA sequencing. What are the new barriers that are impeding progress for the next 10 years? And so we're working on that in our vision. And then finally, what are the big projects that need to be done? And we articulate in our strategic vision a number of those really interesting big projects that we are now just getting off the ground. But it's not going to be all about genomic medicine. I don't want you to think that that's the only thing that needs to happen during this decade. And our strategic vision speaks to the need for continuing to generate high quality reference sequences of the human genome, probably for the rest of the decade. Similarly, developing new technologies, constantly characterizing how genomic variation affects genome function, and thinking about all the nuances of genome function. It'll take us decades to fully understand all those nuances. And of course, we have a lot still to learn about the genomic basis of all human diseases, including the remaining rare diseases. We don't know yet what causes those, not to mention the thousands of common diseases that have very complex series of multiple genomic variants influencing. And then, of course, we will continue to implement genomic medicine, but it's all of these things happening that will help catapult genomic medicine forward. The last thing I just want to tell you, and I'll close with this essentially with one more thing to say at the very end, is this year for our strategic vision, we not only put out those domains of responsible stewardship at the forefront of genomics, but we also made some pretty cool predictions about what you might expect in human genomics by the year 2030. And this actually got a lot of attention. We sort of threw this in at the last minute in our strategic vision, and it was right there in this box, and it listed 10 bold predictions. And that it was really compelling, the 10, but we didn't really talk about them much. And in the text, we just called out a little bit about them. You can just sort of see some highlighted sections where we say, but now more than ever, solution to these problems seem to be within striking distance. So towards that end, and with the characteristic spirit of genomics audacity, we offer 10 bold predictions of what might be realized in human genomics by 2020, see box 5. And what we realized when we published this, especially when members of the press were talking to about it, is that there was more enthusiasm for these 10 bold predictions than we had the ability to sort of bring out from the printed page. And so what we did was to create a promotional video that showcases these 10 bold predictions. And I want to share that brief video with you now.
Genomics, which is the study of all DNA in an organism called the genome, is changing the way we practice medicine, how we manage and treat some previously incurable diseases, and our relationship with society at large. Since the 1950s, double helix and DNA have become household words, standing in for the historic discovery of the 4 chemicals that give us life, commonly abbreviated as A, C, G, and T. Genomics is at an age of innovation and invention like no other, all thanks to how strings of those 4 letters encode the information for creating and operating something as complicated as a human body. Springing forward from the success of the Human Genome Project, a massive reduction in the cost of sequencing a human genome, and the dawn of the CRISPR gene editing era, genomics is leading into an even more audacious future, a future that will define our collective lives for decades to come. But what could this future look like? Here is a summary of 10 bold and fantastical predictions made by experts at the National Human Genome Research Institute and the genomics community for the next decade of human genomics. While most are unlikely to be fully attained by 2030, achieving even one or more of these by that time would have lasting impact for science, medicine, and society. Prediction 1. Sequencing and analyzing a complete human genome will become commonplace for any research laboratory. Prediction 2, the role of every gene in the human genome will be known. Prediction 3, environmental influences on our genome will be routinely used for making predictions about health and disease. Prediction 4, genomics will no longer use social constructs such as race in human research studies. Prediction 5, student projects involving the study of millions of people's genome sequences will be regularly featured at school science fairs. Prediction 6, genomic testing will become commonplace in medicine, just like a standard blood test is used now. Prediction 7, it will be readily known if a given letter difference in a person's DNA is clinically important. Prediction 8, a person's complete genome sequence will be available on their smartphone in a user-friendly form. Prediction 9, advances from human genomics will benefit all of society and not just a few. Prediction 10, genomic discoveries and technologies will help cure more genetic diseases than ever before. How do we get there? The National Human Genome Research Institute is committed to leading research to make such predictions a reality. Learn more about the Institute's 2020 strategic vision for the future of human genomics by visiting genome.gov/2020SV. Dive deeply into a field that continues to transform medicine and will hopefully continue to bring curiosity, purpose, and compassion for people everywhere.
So that's the story I wanted to tell you. I also want to invite you to please stay connected with myself or with the Institute. There's a good landing page at genome.gov/stay connected where you can learn a lot about what you can find on our website. If you're interested in staying in touch with me, you could do it either through a monthly newsletter or through social media, such as through, I guess, what is now X and also LinkedIn. So again, feel free to follow me or to subscribe to my newsletter to visit us on genome.gov. And I will stop there, and I will have left some time for questions. So I'll stop sharing my screen and happy to take any questions.
Dr. Green, that was fabulous. Thank you for sharing that video. You've given us a vision for an NIH institute that really leads the way in the genomic medicine field. And you've generated a huge flurry of questions, so we'll try and get to some of them. And so appreciating you've given us the bold predictions from the Institute. There are a couple of specific examples, perhaps. So, for example, do you think cell-free DNA will be used for screening for cancer? And when do you think that will become standard? And then, of course, will it be reimbursed? And will it be reimbursed better than, will there be parity versus the general genetics cases that might benefit from genomics?
So you know, all those are great questions. I sometimes joke that for a long time it was a lot of fun to be a genomicist because it was, you know, we got to eat at the kids' table where there wasn't as much adult problems to deal with. It was just sort of fun and crazy, and we could sequence genomes and not have to deal with, you know, what adults have to deal with. And these were great questions, like who's going to pay for this, and is it going to be equitably paid for? And are we going to have the same old problems we always have in medicine, where there's, you know, advances but they're not perfect and some people are left behind? You know, these are all things we're working on, and it's new for us, although it's not new for the medical profession, and we're not unique. We have to suffer from the same kind of challenges that and it's interesting to watch different countries grapple with genomic medicine because their different medical systems are more amenable or less amenable. No one's perfect, and so it's all different. So I'm hoping that this will become mainstream. I think there's a lot. I am not a cancer geneticist. In fact, cancer's gotten so specialized that I wouldn't even dare want to make too many predictions or too much expertise. My understanding is they absolutely already are starting to screen more and more, certainly for post-treatment surveillance for some types of cancer. But, you know, for some screening modalities, this is, I think, within striking distance. But, you know, I wouldn't say it's super mainstream quite yet, but from what I hear of the clinical studies going on, there's a lot of optimism for in some cancer domains, this might become mainstream.
Awesome. So another interesting perspective. So many conditions are actually a mix of genetic and environmental factors, right? So how does genomic medicine seek to sort this out? And in that context, for example, becoming standard, the whole genome sequencing of newborns or of just humans in general, or particular groups that may be at risk, how do you incorporate the environmental impact?
Yeah, so I'm a genomics person, so that's why actually I choose my phraseology of genomic medicine, but I tip my hat and say, of course, genomics is only part of the equation. It is why I'm a huge fan of precision medicine, and I'm particularly a huge fan of the precision medicine initiative that President Obama kicked off, which is now exemplified by the All of Us research program here at NIH. It's a trans NIH program. It has a major genomics component, but it is a major program to also capture a lot of other information about individuals, including physical and social factors in people's lives, coupled with their electronic medical records. And I think that's how we're going to crack the nut. I think we need these very large population scale data sets that have both genomic and non-genomic information. And we need a lot of really talented data scientists to help us crunch that data and tease out these contributions. And absolutely right, for the most common health conditions that fill hospitals and clinics around the world, it's not going to be all genomics. It's going to only be a piece of it's going to be genomics, and we need to tease these things out. Among the many things that I think was sort of buried in the bold predictions, and certainly as other place of our strategic vision, just like a week ago, we launched a new program called Multi-omics of Disease, where we are going beyond just genomics and thinking about other omics factors, some of which are readouts of the environment. I mean, epigenomics, for example, I mean, our DNA gets decorated in part because how our bodies are influenced by in the environment. That is a readout of the environment. Or think about microbiome work, which is another, it's our most intimate environmental influences, these microbes that live in us and on us. You know, I don't think of microbiome work as genomic work. I think of it as environmental work because it's an environmental influence on the human body. So there's a lot of excitement, and we're invested in this, but so is the NIH, not only in the All of Us program, but almost every institute is thinking about this.
Awesome. And so from where you sit at the NIH and the focus on genomic medicine, so a couple of questions about pharmacogenetics.
Yeah.
Someone's pondering why CVS and Walmart haven't caught up. It's not even in their database that there might be genomics in play. And what is your speculation about why it's not like utilized as a standard of care? Do you think there's just never not enough convincing outcomes research to get the healthcare system to engage?
Yeah, we've often convened the experts, and if you convene 10 experts, you get 20 opinions on exactly why. So I think some of it, first of all, it's probably more complicated than we thought it was going to be. That's the first thing. The second thing is, I think we have got to have more convincing evidence to get it paid for, because if it doesn't get paid for, then physicians, they just don't want to deal with it. It's another hassle. We also have to get it into the main workflow. It has to be push button. It can't, it's got to be a situation where someone's going to put in an order for a medication, and when they go to put it in, something pops up and says, you know what, that's not the best medication. Or if it's not that pop-up, it's a pop-up that says, you really want to choose between these three, but you won't know which one to choose unless you get a DNA test. And if the patient doesn't already have a DNA test, then maybe you should go get one. But if it's not that convenient in their workflow, they're not going to do it. And so, and if you don't have convincing evidence, they're not going to do it. By the way, in your earlier question, one of the series of questions was, should we just sequence all newborns? And one of the things that comes up is, as you know, would it make sense at some point to have everybody have their genome sequence available so that the medical record would know it immediately, so you wouldn't have to then go through the extra step of getting a DNA test. You would already have every pharmacogenomic signature that you need to know for any medication. That at the moment is probably a fantasy, but maybe we'd be there in 10 years. I don't think it's and we could have whole separate debates about whether every newborn should be sequenced. I and many of my colleagues have the attitude it's not a matter of if, it's a matter of when, that eventually there'll be a tipping point. We'll say it's just worth it. Let's just do it if the parents want it. And maybe not everybody will want it for their child, but I think when there's enough medications that you say that'll influence which medication we give your kid, I tend to think parents are going to just want to get it and have it in their kid's medical record.
All right. There's an interesting, the other end of the spectrum there. Do you have any thoughts about post-mortem genetics? Something I wouldn't have thought of before. Persons who died of complex medical issues, they didn't have an antecedent genomic diagnosis. Would genetic testing potentially inform the family, their peace of mind, closure, that sort of thing?
Oh, it certainly could, but of course, before we even get there, that's certainly the rationale of the All of Us program or other major biobank programs or population scale programs like the UK Biobank is, let's follow people, let's get them and then let's follow them for decades and let's have lots of it's one thing to have a patient when they pass away, you have their information then and you get their DNA then. But imagine if you had their DNA and all their medical history along the way, you might be able to use that information when it's powered by millions of people, not just by a few people, to get some insights about what worked, what didn't work, what was the progression of things, et cetera, et cetera. So certainly, I think, increasingly, you're right. I mean, I think we will want to, if for those who want to know, and not everybody wants to know, it will be having sequencing done in case of an individual might be part of a standard pathology workup, a postmortem.
Okay, and one final question, because I think it's an interesting area, which is what about the direct-to-consumer pharmacogenetics testing, other testing? Where does that have a role? Is it?
Yeah, and I hinted at it earlier when I talked about polygenic risk scores. So, I am very fond of how much the 23andMe's and, Ancestry is even easier to deal with because Ancestry is a recreational thing. It gets a little dicier when you're dealing with companies that are offering health information without a physician involved, because of course there could be overhype and there could be mistakes made by patients not interpreting it correctly and not having a professional guiding them. But I give all of that industry a lot of credit for helping to raise awareness and excitement about genomics. And I'm of the belief that anybody who can get excited about this, we're going to benefit from it. Because as genomics becomes mainstream in medicine, and the more people just understand the fundamental language of it, when their mother or their father, their sister, their brother, their child goes to a doctor and a genomic language is being spoken, it won't be totally foreign to them. So I want everybody's boat to be raised, and some of these direct-to-consumer things have drawn them in. What makes me nervous is when they get out over their skis a little, when it's, it's a little ahead of the data. I think that's gone on a little in the past. I hinted at maybe going on a little now with polygenic risk scores. I certainly don't like the companies that promise to test your child and tell you what sport to put them in, or to test you to tell you what's the best thing for you to eat, or what's the best person for you to marry, and so on and so on. That's to me, that starts to realize a lot of these social genomics things make me very uncomfortable, and they're very money oriented. But but I think with time, I also believe though that people should do what they should be able to do, and as long as it's not forced at them, and as long as it's not misleading. And so, I think there's a place for them in this ecosystem. I always feel better that if it involves healthcare, to have health professionals involved. But I don't think we can't put the genie back in the bottle. So I'm just trying to figure out the best way for us to all coexist in a productive way and not a negative way.
What a fabulous way to end this session. Thank you so much, Dr. Green,
Absolutely.
for your presentation today and very thought-provoking information.
Nice to meet you.
For more information, visit mountainstatesgenetics.org/contact. This activity was brought to you by Mountain States Regional Genetics Network. Please enter this code in the evaluation survey in order to claim genetic counseling, CEU GS23-10.